As concern grows among environmentalists and consumers about micro and nanoplastics in the oceans and in seafood, they are increasingly studied in marine environments, say Baoshan Xing at the University of Massachusetts Amherst and colleagues in China. But little is known about the behavior of nanoplastics in terrestrial environments, especially agricultural soils, they add. They state, our findings provide direct evidence that nanoplastics can accumulate in plants, depending on their surface charge. Plant accumulation of nanoplastics can have both direct ecological effects and implications for agricultural sustainability and food safety. Both positively and negatively charged nanoplastics accumulate in the commonly used laboratory model plant, Arabidopsis thaliana. He says, our experiments have given us evidence of nanoplastics uptake and accumulation in plants in the laboratory at the tissue and molecular level using microscopic, molecular and genetic approaches. We have demonstrated this from root to shoot. Details are in Nature Nanotechnology this week. Xing points out that nanoplastic particles can be as small as a protein or a virus. Weathering and degradation change plastics' physical and chemical properties and imparts surface charges, so environmental particles are different from the pristine polystyrene nanoplastics often used in the lab. After 7 weeks, they observed that plant biomass and height were lower in plants exposed to nanoplastics than in controls, for example. Nanoplastics reduce the total biomass of model plants, Xing adds. They were smaller and the roots were much shorter. If you reduce the biomass, it's not good for the plant, yield is down and the nutritional value of crops may be compromised. He adds, we found that the positively charged particles were not taken up so much, but they are more harmful to the plant. We don't know exactly why, but it's likely that the positively charged nanoplastics interact more with water, nutrients and roots, and triggered different sets of gene expressions. That needs to be explored further in crop plants in the environment. Until then, we don't know how it may affect crop yield and food crop safety. The team also analyzed seedlings to investigate sensitivity of the roots to charged nanoplastics. Exposed for 10 days, seedling growth was inhibited compared with that of control seedlings. Regardless of the surface charge, Arabidopsis can take up and transport nanoplastics with sizes of less than 200 nanometers, they write. Further, in this study, we mainly demonstrate that the pathway of uptake and transport of nanoplastics in root tissues differed between differentially charged nanoplastics. Quote. Thanks for watching. Do you like my video? Then show me your love by pressing like button. Please subscribe my channel and hit bell icon for new technology updates. Bye bye.